Do you remember some of this lab equipment? I'll look through it really quick and see if there's anything you can or can't identify. But A is a petri dish, B is a microscope, C is a beaker, D is a microscope slide, and E is a hot plate. These are all common things that we would use in a biology lab. Anytime you're doing a lab or you want to conduct an experiment, it's going to start with some observation. You notice something, whether you smell it, see it, hear it, taste it, touch it. You notice something. Like, for example, you might notice that your left hand is really fast or that you seem to have slow reflexes. So that's the observation. That's where you want to start. You want to turn that then into a question. For example, which is faster, my left hand or my right hand? And then you might ask another question, are my reflexes slower because I'm distracted? These questions then can become a hypothesis. Once we get a hypothesis, we can design our experiment and we can start to test it. A hypothesis is just a testable statement. Now it's helpful to use what I like to call my magic words. So it's going to start with an if, and then it's going to have an is related to, and then it's going to have then. So something's going to happen as we manipulate those variables. So for example, I noticed that caffeine might affect the heart. So the question is, how much caffeine can affect the heart? I turn that into a hypothesis. If caffeine consumption is related to heart rate, then the more caffeine, the faster the heart rate. Now, it doesn't matter if my hypothesis is correct. It just matters that I can test it. So I could say the more caffeine, the slower the heart rate. That's fine, but I'm just trying to make a prediction, what I think is going to happen here. In this case, I think that caffeine is going to increase the heart rate. From what I know about how caffeine makes people hyperactive, I'd imagine it speeds up the heart rate. Another thing I might notice is a connection between chocolate and pimples. Again, my hypothesis doesn't have to be correct, but I'm trying to make a prediction based on my observations. So it seems like people who eat lots of chocolate, maybe they have lots of pimples. So if chocolate consumption is related to pimples, then the more chocolate, the more pimples. Now that I have that hypothesis, it helps me to design my experiment. Because I said the more chocolate, then I know I'm going to have groups that have increasing amounts of chocolate. So whenever I set up this experiment, I want to have a control group. That is a group that doesn't get anything or gets a placebo. And then the other groups, the experimental groups, are going to get increasingly more chocolate. So group A gets 1 bar a week, group B gets 2 bars a week, and group C gets 5 bars a week. So at the end of a week, I can see which group has the most pimples. And I can carry this experiment on for maybe 4 weeks. Another hypothesis might have to do with light and photosynthesis. So light impacts the rate of photosynthesis. That's my observation. Plants tend to grow better in sunlight. So if the amount of light is related to photosynthesis, then I could say the more light, the taller the plant, or the increase in photosynthesis, something along those lines. And then I can begin to test it. I could have 2 groups. One group of plants is in the sunlight, the other is just in the dark. And I could conduct that experiment. Or I could conduct an experiment where I have increasing amounts of light. So I need some sort of control, a plant that just gets a normal amount of light, and then other exactly the same plant in exactly the same conditions, but they're getting increasing amounts of light. And I do this because I want to isolate a variable. That is, I'm only dealing with light, not the amount of water or moisture, not the type of soil, not the size of the pot. I want to just isolate the variable for light and see the connection. So to understand that a little bit better, if I were to do that experiment, I need to understand some more about photosynthesis. So I might research something like this. And I find that photosynthesis just means I take carbon dioxide and water, and that plant is going to turn that carbon dioxide and water in the presence of light into sugar and oxygen. Now, what I can't really measure very well is how that process works. I could measure the amount of sugar that the plant is producing, or I could measure the amount of oxygen that the plant is producing. The problem with measuring the amount of sugar is that plants use that sugar to grow. They're increasing in their size or their mass. So I might want to use the amount of oxygen released because that's a waste product of photosynthesis. To do that, I might, for example, put the plant underwater and count the amount of bubbles. That bubbles will indicate the oxygen given off. With an increase of oxygen, I can infer that the process of photosynthesis is going faster. So here would be an example. I take plants, I put them underwater. One plant is 5 meters, the next one is 15, and the third one is 30. Now, light has a hard time penetrating that deep underwater, so I'm measuring how the plant is responding this process of photosynthesis by the bubbles that it's producing. And I'm predicting that the plant closer to the surface is getting more sunlight. It should have more photosynthetic reactions. So I'm expecting to see more oxygen released from that plant than I am the one that's really deep in the water. I conduct this experiment and I want to display my results. So we've already learned a little bit about sharing your results with graphs. So I'm going to use a bar graph or a line graph or a pie chart in order to display my results and communicate those results with other individuals who might be interested. Then I'm going to come up with some conclusion that relates back to my hypothesis. I might say something like, in my experiment, I found the more plants are exposed to light, the more they photosynthesize. And I put that conclusion out there, and other individuals can replicate my experiment and test it, and they can modify that conclusion. As they do, it leads to other things. For example, my hypothesis might prove true, but does it prove true indefinitely? In other words, do plants continue to photosynthesize and increase their rate of photosynthesis as light increases indefinitely? Well, we find that that's not the case. At some point, it starts to level off. But my hypothesis explains some observations. We can then come up with some theories based on these multiple hypotheses that we begin to understand better and that are proven true time after time after time. So we come up with some theory. A theory is still in development. It's still being tested, but it explains several different hypotheses across maybe even multiple disciplines. At some point, a theory might become a law. A law or scientific law just describes how something works. So we might describe it the law of gravity. That is, things get pulled toward the center of the earth. We might also run across scientific facts, things that we refer to as just a true statement. That is, it's a statement of truth. Like, we've observed it, we've seen it, we could test it, it happens that way. We would call that a scientific fact. A principle, then, is just some kind of concept based on facts. So something that we can constantly and consistently look for. We would call that a scientific principle. So when we're making predictions about our experiments, a lot of those are based on facts that we already know and principles that we already understand about science. So we understand that plants need light to grow. Well, that's a principle that we're basing that on. So when we say the more light, the more growth, that makes sense. It's making our hypothesis closer in alignment to the principles that we already know to be true. So when we're designing this experiment with stimulus and response, we want to keep some of these concepts in our mind so we can make a scientific experiment that makes sense.